you can hear me. I'm told that if you talk, I'll be able to hear you. Just as are we good? Or
I just
do we need to? Oh, there's a. Sorry, got it. There we go.
that'll be edited out.
Okay, sounds good. Great. I'm happy to take questions throughout the talk. So if there's something that's confusing, or you think that what I'm saying is garbage, or something like that, feel free to just flag me down and let me know. And happy to chat. So before I get going, though, before I get going, I'm hiring. And so if anyone here online or anyone you know is looking for a postdoc, I'm looking for a postdoc to work on projects that are at the intersection of statistical and population genomics. We're specifically looking at how to apply sort of PopGen models to diverse ancestry data. And so if that sounds like something that's exciting to you, it's a potential to be a joint mentorship opportunity with DocEdge over at QBI. And so let me know. There's information on my website right here if you're interested, or you can just email me directly or anything like that. So back. There we are. Okay, so as we were saying, I'm going to talk about assortative mating. So, we've known for over 100 years that assortative mating is potentially has an influence on our genomes and on genetic studies, but it's kind of been the dirty secret, I think, of statistical genetics. We all just, we know it's there, we pretend it isn't, and we hope that it's not a big deal. And that's what we've been doing for a very, very long time. But in recent years, there have been a handful of papers that have come out that have said that, well, let's slow down a bit and let's actually look at how assortative mating may be affecting the types of analyses we do and how it should affect the interpretation of the studies that we're doing, because it's maybe not so straightforward. During this time in the more recent past, even back down to the 70s, there's been a lot of theory work that's been done looking at how assortative mating can affect our studies, like genetic correlations and genetic variants and things like that. So in principle, you could take all of this nice theory work that's been done and apply those principles to then do corrections for our genetic parameters that we estimate. The problem is that all of this theory requires strong assumptions about things like the genetic architecture, how people are sorting, whether or not we're in equilibrium, how long we've been sorting under the current mechanism. And so, trying to just use the existing theory to do model-based corrections is just not going to be feasible for the data that we work with, human data at least. And so what I'm going to present today is an approach that allows us to estimate the fraction of genetic variance or correlation that's due to assortative mating without relying on these strong modeling parameters. I'm also going to present a correction to Mendelian randomization studies that corrects for long-range LD due to assortative mating as well. So that's kind of the direction that we're headed. So why is assortative mating a problem? Well, first I'm going to talk about what assortative mating is. I'm going to use a very broad definition of assortative mating. So assortative mating, I'm going to say, is just anything, it's a kind of observational descriptive statistic that's any kind of correlation between mating pairs. And so this can arise due to, I think, what we by default think when we think about assortative mating, where we actually have people who are mating on observed phenotypes. Tall people are more likely to pair up with other tall people and things like that. So you could have direct sorting. But you could also just have indirect sorting through other mechanisms, including just plain old population stratification. It may be that people are from regions that are more healthy, and therefore you see a correlation of mate health just because they're living in regions where there's kind of different average levels of health. And so both of these things are going to have the same impact on the genome, and I'm going to call them all assortative mating altogether. There's also different ways you can also think about assortative mating. You can have single trait sorting, where you have, for example, tall people more likely to mate with other tall people, so matching on the phenotype. You can have sorting across traits, where maybe you have tall people who are more likely to mate with more athletic people, where there's correlated across phenotypes. But more realistically, we could have something that's I'm calling multi-trait sorting, where people are sorting on a large vector of characteristics that all you could maybe treat it as a sort of a combination of single plus cross trait sorting, but a more complex idea of what sorting actually is. So then why is sorting a problem? Well, sorting creates long-range LD in the genome. And so how does this long-range LD show up? Well, here's a little toy example. So imagine that people are sorting on height, and we have two alleles, A and B, which both increase height. But they're on different chromosomes. So in a randomly mating population, these two things should be uncorrelated. Well, if there's a person, a child, and they have a copy of allele A, then they must have gotten that allele from one of their parents. So they have a parent with allele A, which means that parent one is going to be more likely to be a little taller. if there's assortative mating on height, that means that the other parent is also more likely to be a little taller. And if they're taller, then it means that they have to be taller for some reason. And so they're more likely to have any allele that's height increasing, including this allele B. And so if parent 2 has a copy of allele B, then they're more likely to pass that on to the same initial child. And so these two SNPs that would be uncorrelated in a randomly mating population can now be correlated because people have this correlated height, because people are sorting on height. So that's kind of the underlying mechanism of how correlation can happen. As you see, some of these steps, you know, the increase in likelihood of having allele B is just little, right? And so the correlation might be slight, but there's a lot of SNP pairs where this is going to happen, and so this can add up to a lot. So then let's think about how this applies to the genetic parameters that we think about. So first, genetic variance. So what I have here is that imagine we have some phenotype, Y, and it's the sum of, I've broken up the genome into chromosomes. And so GKY is the genetic component for chromosome K for phenotype Y. And then we have epsilon, which is the residual, right? And then if we take the variance of just the full genetic component, that separates out into a bunch of within chromosome terms and a bunch of cross chromosome terms. Now, if there were random mating, these cross chromosome terms would all be 0. However, when they're sorting, as I was talking about, these terms all of a sudden become non-zero. You know, if people are sorting on trait Y, these things will generally be positive. And so you can see that, you know, due to sorting, genetic variance can increase in this way. You know, correlation is going to be the same sort of story, right? If we have the genetic component for some trait Z and the genetic component for some trait Y, and then we separate this out into the within chromosome terms and the cross chromosome terms, we have all of these terms here, which the cross chromosome terms, which would be 0 in a randomly mating population, but are going to be non-zero in a potentially non-zero in a sorting population. I want to highlight one kind of important but sort of technical point. When I talk about genetic correlation here, you may notice that I'm actually taking the correlation of the genetic factors. So Border calls this the score correlation because you could think of this as the genetic score. You could alternatively think of genetic correlation as the correlation of the effects of the genetic variance. So Border calls that the score correlation. Whenever I talk about genetic correlation, I'm always referring to the score correlation. The reason for that is effect correlation is not affected by assortative mating. But also, if you have studies that use, I'm going to use the term polygenic index through this talk, but it's the same thing as a polygenic score, if you've heard that. Score correlation is what is relevant for studies that use polygenic indexes. And so anything I say about genetic correlation in this talk is going to hold for polygenic index studies as well. So we see that these are also going to potentially be influenced by assortative mating as well. Now, Mendelian randomization is a little bit different. And for those of you who haven't thought about MR recently, here's a quick primer. So imagine that we have our same traits, Y and Z, and the causal effect of Z, I'm going to just assume is equal to gamma. So, however, because in just normal data, I'm using the causal effect gamma here, this epsilon will not necessarily be uncorrelated with Z. And so if we were to just do a simple OLS regression of Y onto Z, then this will give us a biased estimate of gamma because Z is correlated with the epsilon here, right? So we can't just do OLS. And so MR folks say, well, let's find a SNP that, and we're going to take the covariance of both sides of this expression with respect to SNP X sub J. So we do that here. This is how it expands out. And then we're going to assume that this SNP is uncorrelated with the epsilon term. And what that means sort of intuitively is that the only way that this SNP is correlated to the outcome variable is through its correlation with the treatment Z. And so, so if this is equal to 0, then we can solve for gamma. And that gives us this ratio here. I'm going to multiply or divide the top and bottom by variance of X. And so you notice now in the top, I have an OLS regression parameter. This is the GWAS regression of SNP X onto Y. And so I'm going to call that beta YJ divided by beta ZJ, right? So this is just, what you may see when you're doing MR, it's just the ratio of GWAS parameters, and this is where it comes from. I'm going to call this ratio here the MR ratio. So in general in MR, we don't just have, just one thing. So I just want to highlight this assumption here is key though, right? So the key assumption of MR is that this covariance is equal to 0. That's called the exclusion restriction. Now, it may be the case that the exclusion restriction is not met for a particular SNP. We often don't know what SNPs do. the genome is complicated, SNPs do a lot of different things, and we don't know the pathways through which they operate. But however, an advantage in genetic data, potentially, is that we have lots of SNPs, that, there are lots of potential pathways, and there have been a lot of methods that have been developed where we can, calculate these MR ratios for a lot of SNPs, and then apply a method that will be a consistent estimator of gamma as long as the exclusion restriction holds for like some subset. So like median MR, if you've heard of that, requires that at least half of the SNPs satisfy exclusion. Or you can do mode MR, which is that you need, that's more technical, but they say like a plurality. You need more SNPs that satisfy exclusion than don't satisfy exclusion. There's also approaches that say, okay, well, it's okay if some of them don't satisfy exclusion as long as the violations of exclusion are uncorrelated with other stuff. So there's millions of MR methods that that assume that different things about different sets of SNPs. But what I want to point out here is, again, this coefficient here, the estimate of an MR estimate is just this ratio of these covariance terms. But if I were to break down Y into the chromosome-specific components, we see that this X, if there's assortative mating, is going to be correlated with the genetic component everywhere across the genome. And so a SNP only satisfies the exclusion if it satisfies exclusion and the SNPs it's correlated with also satisfy exclusion. And so when we think about what assortative mating is going to do, it's making this correlated with all SNP, in fact, all genetic variants in the genome. And so the consequence of this is that a SNP will only satisfy exclusion if there's assortative mating, if every genetic variant in the genome also satisfies exclusion. So it starts with this sort of assumption, which is perhaps a bit tenuous at first, and it turns it into something that's totally implausible, because we don't know anything about all these genetic variants, especially the ones that we don't see. And so assortative mating is potentially a disaster for MR studies. Okay, so this is the problem. So I'm going to now talk a little bit. So one of the parts of this project, we extended the theory that like I said, there's a lot of theory that exists already. Generally, it assumes pretty specific models of sorting. There's also a lot of simulation work, which just by nature, simulations are pretty a narrow view of what goes on, because you only know about the models that you can simulate, that parameter space. And so we've developed just a little bit of theory that extends the sorting theory that exists into a more general framework. And so let me talk just briefly about what we've done. So we start actually with a model of assortative mating that's from the economic literature, since that's where I'm from. And so the idea is that what we have to assume is that sorting can be defined by some latent, unobserved parameter, which is potentially a complex function of a million different things, the cubed root of the log of height times, you can just make up whatever you want. And I'm assuming that there is this S thing that defines sorting, and it's based on the things that are observed and unobserved, and maybe some things that are just totally random. I'm also going to allow for this latent sorting parameter to be different for men and women. So it may be that men sort on different things than women. And then the way sorting happens is we rank order everyone according to this unobserved S, and then people just match according to who's closest to them in terms of this latent S parameter. Okay. So this is quite general. The sense in which is not general is that you could imagine situations where sorting isn't defined by a univariate thing, and you actually need multiple dimensions to define sorting. And so we can't capture this, but this is more general than the existing theory, which assumes that it's just people are sorting on one thing, or maybe cross-trait sorting on two things. So then how does this latent sorting parameter relate to the observed things that we're interested in studying? So imagine that we knew what S was. You could imagine regressing S onto our treatment trait, Z, and our outcome trait, Y. Or if we're thinking about genetic correlation, Z and Y are two traits, and we don't care which one's treatment or outcome or any sort of notion like that. And so this is just a thing that you can do. It's a statistical exercise. And so that produces these theta terms. And so these theta terms here, you can interpret as how much does, so like, for example, theta Zm is how much does the treatment trait explain how mothers are sorting? And similarly for, theta Ym is how much does the outcome trait explain sorting in fathers? And then there's some residual term, which would be, traits that are unexplained by Z and Y. So we're still very general. I'm importantly not going to make any assumptions about the genetic architecture of any of these traits. I'm not going to make any assumptions about whether things are pleiotropic or not. You know, we could have, you know, traits with thick tails or crazy bimodal, I don't really care distribution that you want. I'm making no assumptions, but so far the only thing I've assumed is that this is univariate. Okay. So I'm going to put up a table that has a lot of stuff on it that I don't want you to think too hard about. This is just to illustrate that just under the simple model, that's all you need to then go through, and you can actually solve what's going to happen to LD and then what's going to happen to all of these genetic parameters that we're interested in. So I listed here, for example, you could just assume that people are just sorting on Z or just sorting on Y, or maybe there's some kind of cross-trait sorting, and then calculate how much is the genetic variance going to increase? What's the genetic correlation going to be? What's the GWAS bias going to be? So this is messy. This is actually what it is after only one generation of sorting. And you can see already, this is kind of a mess. But one thing that is interesting about this is that even though this is kind of a mess, this is only all of these cells, and even in the very general case, which I didn't want to put up because it filled the screen 3 times, is only a function of these theta parameters, which are the parameters that define the relationships of sorting to the traits we're interested in, and then things like, you know, the heritability and the genetic correlation, the random mating genetic correlation. And so, you know, this highlights that even though complicated things can be happening, things are tricky, but not, you know, arbitrarily tricky. The other thing I want to point out is this column here. So what I've done here is I've said, what is the MR ratio going to be if there's assortative mating, even in these simple cases, if the true effect is gamma, and we're only looking at SNPs that satisfy exclusion in the randomly mating population. And so if MR were unbiased in a sorting population, then this thing would be equal to gamma. And you can see that this in general will not be equal to gamma. This is, I think, especially important because there is literature out there that claims that Mendelian randomization is unbiased when people are just sorting on Z or just sorting on Y. And we can see that this doesn't hold in general. And this is something that we catch because we've done this very flexible theory versus just kind of sampling in a simulation space. I guess there's one other point I want to make. Notice that all of these have thetas in them, and that's actually the problem for why we can't do model-based corrections. You know, theta is the parameter that we can't easily estimate because it's a function of this unknown latent thing that we know nothing about. And so we can't do a model-based correction, so what do we do? So at a high level, what I'm going to walk you through is a method that approximates how much of the genetic correlation or how much of the MR estimate is due to assortative mating. What we're going to do is we're going to go and we're going to identify how much of these parameters is driven by cross-chromosome LD, and we're just going to remove it. Now, importantly, assortative mating it can influence within chromosome LD, and we're not going to do anything about that. But what I'm going to claim is that because most SNP pairs are cross-chromosome SNP pairs, you know, 96% of SNP pairs in the genome are cross-chromosome SNP pairs, we're going to be able to remove, you know, almost all of the bias due to assortative mating, but we're, you know, in expectation going to miss about 4%. But I'm going to say that's close enough to at least tell us something interesting. And in general, the way that we're going to do this is we're going to use polygenic indexes. So let me walk you through specifically how we do it. So for genetic variance, so notice here I have this ratio. In the denominator of the ratio here, this is just repeating what I showed you before. This is the total genetic variance for trait Y. In the numerator of this ratio is just the variance that's due to within chromosome variation, or within chromosome variance covariance, right? And so the interpretation of this ratio is the fraction of the genetic variance that's due to just within chromosome LD. Now, we don't know the chromosome genetic factor. You know, we don't know G. We try to estimate it, but we don't know. In fact, we do estimate it. That's what a polygenic index is meant to do. And so what we're going to do is we're just going to substitute out all these unknown G's for G hat, where G hat is the polygenic index. So G hat YK is the polygenic index for trait Y for only SNPs on chromosome K. And so if we just take this ratio, that's going to be it will be the fraction of the variance in the PGI that's due to within chromosome variance. And I'm going to claim that that's a fairly good approximation of the total genetic variance for trait Y that's due to assortative mating or long range LD. Now, there's a few things that are wrong with this that you might see immediately. First off, polygenic indexes don't include every genetic variant. And so, there's a sense in which these PGIs are imperfect, incomplete, but they also add a lot of noise because we don't know the effect of every genetic variant. We have to just use estimates, usually based on GWAS summary statistics. And so there's also noise. I'm going to claim that because, well, first off, for the omitted SNPs, if you assume that the long range LD due to observed SNPs is representative of the long range LD in unobserved SNPs, then I think we're still going to be okay here. Where I get a little bit less comfortable is saying, well, there's error in the top and the bottom now. And so that can cause problems. And I'm just going to claim that, well, because it's in the numerator and the denominator, it's going to at least partially cancel. And again, this is going to be imperfect, but I'm going to say this is going to be good enough that it tells us something interesting. And in practice, we have some simulations where it shows that it does pretty well.
Is it also in that, you're assuming you have at least one causal SNP on each chromosome.
Yeah.
And the estimate with one causal SNP on each chromosome is 4. And the estimate with a lot of chromosomes is better with that error that you were just talking about. So there's some sort of minimum number.
Yeah, yes. So you do need some degree of polygenicity for this to work out well. So all the stuff I'm going to show you are going to be traits that generally are quite polygenic. But if we were looking at, something that isn't, it could be problematic. But everything I has it's believed that the chromosome specific heritability tends to be proportionate to the length of the chromosome, I think, for all of these traits.
Okay.
And I tend to work with sort of behavioral traits, and all of those are totally polygenic. So we're definitely fine in that region. So that's how you do it for the variance. And then, I could walk you through the covariance, but it's exactly the same. We do the same kind of thing. We have the within chromosome and cross chromosome covariance. And so, now that we have these K parameters, if we want to know what's the fraction of genetic variance due to AM, it's just going to be 1 minus this K ratio that I showed you before. If we want to say what's the fraction of the genetic correlation, then, correlation has a covariance term and 2 variance terms. And so it's just going to be 1 minus this thing. And so that'll give us estimates of how much long-range LD is affecting these parameters that we're potentially interested in. So we've done a bunch of simulations. I'm not going to talk in detail just due to time about everything that we did, but we see here we looked at a handful of different sorting mechanisms, single trait sorting mechanisms, cross trait, multi-trait, and then we also looked at a handful of different joint genetic architectures between the 2 traits. And so the blue bars are the actual genetic correlation in generation 1 versus generation 4. And so, if you see that the blue bar in generation 4 is higher than the solid or than the dashed line, that means that genetic assortative mating is inflating the genetic correlation. We see that generally happens such in a handful of cases here. And the gold bar is the genetic correlation estimate that we get once we do the adjustment that I've shown you before. And so you see that the gold bars pretty much always get you almost all the way back down to the dashed line. So when I'm saying that it seems to do pretty well, but not totally perfect, that's the sort of order of magnitude that I'm in. So it does pretty well. So let's talk about Mendelian randomization. So it's going to be a little different. Instead of correcting the Mendelian randomization estimates directly, we're going to correct each GWAS coefficient, each GWAS association, just one SNP at a time. And so, in the denominator here, this is the covariance between SNP X and or SNP J and the genetic factor for Y. And then, across all chromosomes. And in the numerator, we have the covariance between SNP J and the genetic factor just for the SNPs on the chromosome that correspond to SNP J. And so, these G's we don't know, we replace them with the polygenic indexes. And so this gives us a correction factor of, how much of the GWAS association for SNP J is driven by long-range LD. And so we can just multiply the observed associations based on actual data by our correction factor, and that gives us adjusted betas. So then we can use those betas and just plug them into any MR algorithm that we know about. One tricky thing is that these K ratios, in this case in particular, is a ratio of two very small numbers. And as you might expect, the ratio of two very small numbers tends to be really unstable. Sometimes this correction causes these associations to jump up to things that are just totally implausible. And so what we're going to do to deal with that is we're going to use median-based MR for our actual kind of MR method, because this is a method that's more robust to the outliers that may arise due to the crazy instability of this adjustment. And so specifically, our adjusted causal estimate is going to be the ratio of the adjusted GWAS coefficients, and we're just going to take the median of those for just the set of SNPs that are statistically significant for the treatment trait. So this is just the classic median MR estimator, except we're using the adjusted betas here in the ratio. So as if you were paying really close attention, you notice that I am not calling this an assortative mating adjustment. This is just a long-range LD adjustment. And so the reason for that is because there's a selection problem. So this is another set of simulations that I showed here. We have both the median and the mode MR estimates for the simulated data. And we see here that these are biased downward, but when we do our adjustment, it gets us closer. These are biased upwards, but when we do our adjustment, it gets us closer. But we have this case here where there's a lot of bias, but once we do our adjustment, we're only getting halfway to the truth. And so where does that come from? And the issue is that there's additional assortative mating induces selection bias in addition to the bias in the GWAS coefficients due to the set of SNPs that are selected. How am I doing on time? Just fine. So let me walk you through how this happens. So what I've plotted here is each one of these circles corresponds to a SNP. So I've simulated a bunch of SNPs that have different effect sizes, and the point here is a single SNP, and its position is, its horizontal position is its effect on the treatment trait, and its vertical position is the effect of that SNP on the outcome trait. So this SNP here has an effect of +2 on the treatment and -2 on the outcome. Right? This one here has a 0 effect on both the treatment and the outcome. The blue arrows here correspond to after 4 generations of assortative mating, what's going to happen to the GWAS coefficients in expectation. And so you can see that because, and specifically, this is under a model where people are sorting on just the outcome trait, and that there's a genetic correlation between the treatment and the outcome of 0.5. And so you can see that there's a lot of bias upwards because, or away from 0, because people are sorting on the outcomes is maybe what you would expect. But there's also a little bit of horizontal bias, or I guess pretty substantial here too, to be honest, because it's pretty extreme simulation. But there is some bias also in the treatment GWAS due to the correlation between these two traits, even though people are only sorting on the outcome directly. Now, the important thing I want you to notice is that the horizontal bias, or the bias on the treatment GWAS, the magnitude of that bias is a function of its vertical position, or a function of the effect of that SNP on the outcome trait. And so, this SNP here has a big effect on the outcome, and its bias on the treatment is much larger than this SNP here, which has no effect on the outcome, and therefore there's no bias at all on the treatment GWAS. And so this is going to cause problems. Let me show you why. So now instead of just plotting 25 SNPs I showed you before, this is all of the SNPs. I've oriented the SNPs so that they all have positive effects. But this is just a set of SNPs that have a correlation of 0.5. This dashed line here is, I have a line that's going through the origin, and I plotted it such that half the points are above the line and half the points are below the line. And the slope of this line here is the median MR estimate. It's just another way of portraying median MR. So half above, half below. So after 4 generations of sorting, the GWAS coefficients are going to stretch in the way that I showed. So they're mostly stretching up, but they're also stretching outward as well. I guess these ones down here are stretching that way. But this is the stretching that occurs due to 4 generations of assortative mating. And so if we just take the set of SNPs that are have 50% power to be detected in a GWAS of some fixed sample size, those are just gonna be SNPs that have an effect size greater than some fixed amount. And so I'm going to just highlight those SNPs and draw a line that passes through those points so that half of them are above the line, half of them are below the line. And so the slope of this line here is the median MR estimate for SNPs that, after 4 generations of sorting, doing the selection criteria that is always done in Mendelian randomization. So then let's say we correct this. We have, let's say we correct it perfectly. We're able to put all these SNPs exactly back to where they were in the randomly mating population, but we only use the SNPs that were captured in the sorting population GWAS. So what's going to happen is these SNPs up here are going to shrink back a lot. And these ones down here are only going to shrink back a little. And as a result, now I've drawn this line here going through these remaining points. You can see that it's not a vertical line that defines what's included in the MR analysis and what isn't. It's this angle. And as a consequence of that angle, when we draw this line through the points of the half are above and half are below, we don't get back down to this, slope of 1/2 line. And the distance between the gray line and the black line is selection bias in MR. So, we have some big, from here to where it was up here is biased due to long range LP, and from this line to this line is biased due to selection. So these two sources of bias when there's assortative mating in MR studies. So that was just a simple case. It turns out that if you use our theory, you can actually work out what the conditions are where you would generally expect to see bias. And as you may have inferred based on how I was presenting that previously, the condition that's necessary for there to be selection bias is that the bias on the treatment GWAS needs to be a function of the effect of that SNP on the outcome trait. And so we can go through our theory and figure out, well, under what conditions will that be? And it turns out this is the condition here, that if this expression here is non-zero, there will be selection bias. Let me talk you through these different terms. So as you recall, potentially, these thetas are how much, so theta y is how much trait y explains sorting in the population. And we notice that these are cross-trait products. And so this is theta ym, theta yz. So that means high Y people, high Y mothers are sorting with high Z fathers. And so this expression here, the left expression corresponds to cross-trait assortative mating. And then you notice here it says theta y, which means that this is the situation when people are sorting on just the outcome, which is what I illustrated in the previous slide. So there's the potential for selection bias if there's either cross-trait sorting or sorting on the outcome. And so if you're in a situation where you expect there to be either cross-trait or outcome sorting, then MR is potentially problematic due to selection. Great. So I'm going to go through the application. We talked about all this theory. So the specific application that I'm going to talk through here is about education and health. The reason I'm talking about these is because I do a lot of my research in education, but also there's this very strong empirical relationship, a phenotypic relationship between education and health. People who are more educated tend to be more healthy. And this phenotypic relationship is also backed up by genetic evidence. And so there's a strong genetic correlation between education and a lot of health outcomes. The polygenic indexes for education tend to be very highly predictive of health conditions. And so, however, as I've been showing before, those correlations and PGI associations are potentially just driven by assortative mating. Also, there's a huge literature on the effect of education on health conditions. I just did like a Google Scholar search for MR analyses since 2019. So this is the last few years. There were just hundreds of these studies in literature. I just took a few of them to show that education has been shown to have a significant MR estimate of the effect of education on health, stroke, lung cancer, healthy lifestyle, Alzheimer's disease, COVID, you name it. There's just an MR study for everything. Importantly, most of this evidence is just based on observational data in unrelated individuals and therefore is potentially biased by assortative mating. So I'm interested in how well do we expect these to hold up if we applied the adjustment that I presented here. Especially given that education is one of the traits where we observe the highest sorting. I mean, education and height are the two kind of main sort of I guess, there's like skin color and things like that too, but in terms of kind of behavioral phenotypes, education is one of the very highest on which there's sorting. So for the data, we just look at, so far, I guess, we've just looked at 8 health measures. And so asthma, BMI, cigarettes per day, depressive symptoms, ever smoker, neuroticism, self-rated health. And we also look at height, which is not exactly a health condition. I sort of think of this as a negative control. You wouldn't expect education to affect a person's height, especially in the developed country, where since a person's adult height is sort of set by the time they finish their education. So I include that as just sort of a negative control. The data that we use is just the European ancestry subsample of the UK Biobank. For the genetic correlation analyses, these PGIs come from the SSGAC PGI repository. And so they just created a bunch of PGIs in the UK Biobank and a bunch of other samples too, for a lot of different conditions, but including these 8 plus education. And then for the MR analysis, what we do is we take 1/3 of the UK Biobank, we do a GWAS in 1/3 of that sample. And then for the PGIs that we use in the adjustment, we use the PGIs from the PGI repository again, that are just based on the other 2/3 of the UKB and other published data. So first, let's think about how assortative mating affects genetic variance and correlation. So here, the gray bars report what we estimate how much of the genetic variance for each of these traits is due to assortative mating. And so, starting here, as maybe you might expect, this fraction is highest for education, more than anything else, overwhelmingly more so for everything except for height, which, height and EA are the 2 things that people sort on the most. But we see about 10% of the genetic variance for education seems to just be driven by assortative mating, and a little less than that for height. Otherwise, everything else is quite low. Self-rated health, so this is self-rated health here. Self-rated health is about 5%. Everything else, I think, is less than 3%. So assortative mating may be driving a moderate amount of the genetic variance for education height, but doesn't seem to be driving much of the genetic variance for these particular health conditions. The story is different for genetic correlation, though. So for genetic correlation, there's some variation. It gets as high as almost 40%. This is drink. Did I say I studied drinks per week on the previous slide? I'm realizing that. I don't think I did. That slide's incomplete. DPW is drinks per week. And so, we see that, a large fraction of the genetic correlation between education and drinks per week may just be assortative mating, but everything else is pretty moderate. The average across all of these traits is about 14%. The reason why I think this is actually interesting is because there was a paper that came out in Science somewhat recently by Richard Border and his colleagues, where they argue that a huge fraction of the genetic correlations that we see in the literature could just be driven by assortative mating alone and not due to any sort of pleiotropy or things like that. I think that they, for these particular traits, I looked up actually all the education health associations that they looked at in their paper. And I think on average, they said that you can get almost 30% of the way to the observed genetic correlations just by looking at sorting. Whereas in my case here, it just seems like it's like assortative mating does seem to matter. It does seem to drive some of the genetic correlation, but 15%. It's not huge.
Was that study done on the UK Biobank data, or was it a different?
So that study,
different across population?
Yeah, so that study did not exactly use data in the same way. It's a simulation-based study. So they look at the spousal correlations in, I think, the UK Biobank and also the Danish. So I forget exactly what the other study is. So they try to match the spousal correlations that exist in that data to define how sorting happens in their simulation. And then they simulate data for traits that are not pleiotropic, that have a 0 genetic correlation, but then they model a sorting mechanism that nails the phenotypic sorting of spouses.
Okay.
So it's not exactly an apples to apples comparison. Yeah.
Quick question. So you presented some of your results for the MR were showing how the adjustment can't correct all of the bias because of like selection factor, right? The selection effect. However, for like a polygenic score, that itself, isn't that also constructed post-selection, and so is that accompanied for here also?
Yeah, so I
or
guess it
why do we
depends.
not see that here but
Mhm.
there?
Yeah, so I base this on polygenic scores that aren't, they're not like pruning and thresholding type scores. These are I think we made these with either, I don't remember if they're LD-Pred or PRSCS, but these are genome-wide scores, so there's no
Okay,
selections there.
Okay, okay.
But it's true, yeah. If you were to build your polygenic scores using a pruning and thresholding method, yeah, you might expect there to be some sort of selection there too. I haven't thought through that carefully. That's a great point. So the story is a little different for MR. MR seems to be a little bit more sensitive. So all of these bars is the estimated effect of education on the corresponding health trait. We see in general that, the naive estimate is that education improves health. Not all of these are significant, but in general, they're all in the right direction of, what we, the narrative that you might tell yourself. But the question is, how much of that MR estimate is driven by long range LD? When we do this correction, the average reduction in that coefficient is about 30%. So, at a baseline, MR estimates of the effect of education on health are probably inflated by about 30%. But then, as I said before, our adjustment can only adjust for long range LD. It does not adjust for selection. And so, look at height, for example. Our long range LD adjustment drops us about halfway. Presumably, if we were able to account for selection, it would drop us the rest of the way. And in our simulations, when there was selection bias, the selection bias tended to be of comparable magnitude to the long range LD bias. And so, this 30% reduction is in my head, I kind of double that just based on looking at the simulations, but you can say at best the estimates are 30% smaller, and probably they're much smaller. Given that there's any shrinkage at all, that means that there is evidence of assortative mating between these two traits to begin with. And so we should probably approach these results with general skepticism, in how we update our priors of what the actual effect of education is on these traits. So just a handful of discussion points. So, we did this extended theory. And one of the reasons why I really like this theory again is because, we did this very general model of sorting, and it fills in the gaps that I think are left by all the simulation evidence. There's a ton of simulation evidence in the literature. And some of our theory confirms conjectures that exist in that simulation evidence, and some of it actually disproves it. So for example, Border, I'll do a lot of simulations, and they say it looks like, pleiotropy and other higher order genetic architecture things don't affect how much assortative mating affects genetic correlation. And we do our theory analysis and say, yeah,
Okay,
that's
Coffee,
actually true.
fingers.
It doesn't matter. Oh, is there a question? I thought there might have been a question.
Yeah.
Okay, I will keep going. So that turns out to actually be true. The other one is this, I mentioned this when I went through this in the middle of the slide, but there's this simulation paper here where they say it looks like single trait sorting on just the treatment or outcome will not bias Mendelian randomization studies. However, our theory suggests that actually it can bias these sorts of studies, and the problem is that the simulations just weren't broad enough to capture the cases in which you would expect to see bias in these kinds of studies. So in addition to that, I just talked about genetic correlation and MR and genetic variance and things like that, but you could expect assortative mating to affect a wide variety of types of studies that we look at. Anything that looks at sort of subtle comparisons that are kind of genome-wide comparisons. So for example, there's this paper, Demange et al., where they look at the biological annotation of cognitive abilities compared to the biological annotations of non-cognitive abilities, and they find that they're quite similar. And they say, oh, well, this is interesting. It looks like sort of the biological underpinnings of these two traits, they're driven by the same things, even though these two traits have kind of a low genetic correlation. However, the result that we have would suggest that, you might actually expect that the biological annotations to be the same if people are cross-trait sorting on cognitive and non-cognitive abilities, which sounds sensible to me. Another one is, there's this paper on the omnigenic model that came out in Cell, I guess, 6 years ago now, where they observe that with disease traits, that there's always seems to be sort of a core set of disease-related genes, but then sort of an omnigenic backbone of just kind of low-level traits that seem to be affecting these diseases, potentially due to regulatory mechanisms and things like that. But if you imagine a situation where people are sorting strongly on something that is highly polygenic, like height or educational attainment, what that will do is that will create just a low level of LD throughout the whole genome. And therefore, every SNP in the genome is kind of just going to be picking up a little bit of the effects of actual causal SNPs throughout the genome. And so what looks like might be some sort of omnigenic backbone could actually just be the result of this low level of long-range LD due to assortative mating on polygenic traits. And so I think that, thinking about assortative mating really colors how we potentially colors how we interpret this. And, I could go on and list like a million other potential things, but I think it just means that we really need to think hard about how assortative mating may affect how we interpret the work that we do. That said, when we actually look at genetic correlation for education and health, it doesn't seem to be that huge a deal, right? 10–15% of the genetic correlation there is due to assortative mating. So maybe there is something real happening there. MR studies are more sensitive. And overall, this highlights the need for things like family studies, right? If we look at family where we control for parents or we use siblings, that actually eliminates the long-range LD, at least the cross-chromosome LD. And so, I think that this just heightens the need. I think we're all talking about this already in the field, but we really need these family studies. But we still need to be careful. And one thing I want to highlight is there's a paper where they say, okay, we should be doing, for MR studies, we should be using family data. However, there's not very much family data. And so maybe what we could do is we could pick the SNPs, because we're underpowered to identify SNPs that we should use in our MR studies. So they said, let's go on and we'll use the population GWAS to pick out the SNPs that we want to use in our MR study. And then we'll use the unbiased family-based GWAS estimates to estimate the causal effect. And so that will get rid of the long-range LD bias. However, because we're picking the SNPs based on the population data, the selection bias is still going to remain. And so, just highlighting that as sort of a sort of mating can catch you in ways that are unexpected if you're not very careful. And even with that, I want to say one last thing, because even if we can correct for assortative mating, horizontal pleiotropy is still a concern. Just because it's not long-range LD doesn't mean the exclusion is satisfied for any particular SNP. So I always like to encourage people to be very cautious in applying MR in studies where we don't know what the SNPs are actually doing. So I think that's the main thing. Just thanks to you all for having me. Thanks to my co-authors, especially Marta, who is my research assistant, who I think is online. She's driven most of this work. She's been working with me for the past 2 years, and she's leaving me for grad school this summer at MIT. So I'm very proud of her, but I'm going to miss her. So thanks, Marta. But also to my other co-authors who are Dan Benjamin, Paul Auer, Michael Livermore, and Miles Kimball. And also thanks to all the people who've funded this work. I have my contact information up here in the corner. So if you want to reach out to me directly, feel free. But I think I'm also just hanging out here for the next hour to chat with people who are here who want to chat.
Great. Any questions from those in the room? And
yeah. So there was a recent discussion about alcohol, moderate alcohol, and health. How much does this sort of maybe play a role in that phenomenon?
Yeah. So the only empirical evidence that I have that I guess we have. It seems like the genetic correlation between how much a person drinks and their education, at least, is that seems to be driven largely by assortative mating. So it does seem like sorting could be a big problem. I'd have to look specifically at the application you're talking about, but yeah, drinking behavior seems to also be a trait, and as you might expect, a trait where sorting is potentially a big deal.
Right. So we just need to, studies that are not genetic studies that are just looking at the correlation between, bias and non-happening the overall health, interpreting what seems to be a bias, interpreting that as being due to if the water can be associated with water.
That's a great question. I guess the only sense in which this work would apply to that directly is, if you look at the genetic component for drinking and the genetic component for health, the correlation between those two pieces will be inflated by assortative mating. And so you could imagine assortative mating inflating those correlations. If people are sorting though, you could imagine all sorts of crazy things happening in the non-genetic part of both of those things that is hard to predict. But at least that genetic component, I think we could say, predictably that assortative mating could be driving up that portion. Does that answer your question?
I think so.
Okay, thank you. Yep.
It's a really cool framework, Patrick. In terms of evaluating a polygenic score, right? So like, imagine we run a study, whether or not we do selection for variants, how we construct it, right? So we get some genetic effects from GWAS, we construct a PRS, and we want to get some sense of validation to see if it's predictive out of sample or in a validation cohort. Would, rather than just saying there's a single score for each individual, if you did like the chromosome specific score, right, and did evaluation of that, would that at least adjust for some of this, right? Because there, if you have the per chromosome score, right, like in a linear regression, right, like each of those should be, those estimates are conditioned on the rest of it. And so, at least in the sample, right, would that be adjusting for some of this sort of induced long range LD?
That's interesting. So I guess there is some stuff happening within a chromosome that you might worry about.
Sure, but like similar to what you said.
Yeah, but it's not
I'm certainly not suggesting that as an alternative approach, but just to say for specifically to capture some of the behavior of what you're getting at here, if you want to evaluate the predictive utility or predictive power of the PGS, rather than just saying, all right, everyone has a score, does it predict their phenotype? To say everyone has a chromosome specific score.
I think you're right. So I think that if you were to break up a polygenic score into chromosome specific things and then include all 22 small scores into your regression, and then you take the variance of each of those things and just add them together, and say like, this is actually the effect of kind of within chromosome stuff. I think that's like ultimately an equivalent framework, right? Because if you want to say what's the, because you're ignoring the covariance terms if you're just kind of raw adding up each of those individual things. So
that seems like the wrong thing. Because what the problem is, there's cross chromosome correlated and you're not accounting for it.
Well, I think that's what this would adjust for. Because you would have it would.
But if you put 22 scores in the model, you're going to assume independent effects. You're going to estimate those effects. It makes it better to put all SNPs in the same model, so when you compute the weights for your PRS.
Oh, no. So not computing the weights, this is evaluating after you already have it.
Yeah, so like
you're instead of saying, here's everyone's score, does it track with their outcome? You have a regression framework that says, the additive contribution of those 22 scores predict the outcome.
Yeah, because the way that these are often interpreted, right? So you do a regression of the like asthma risk onto the education polygenic score, and you have some coefficient, and he's like, oh look, the education, like they must have some common biological underpinning that corresponds to what this is explaining. But there's all these cross chromosome terms, and so if you did this thing yourself, like you were talking about, where you break it up by chromosome, then I think that's just going to be, it might even be, you probably would show that it's an equivalent framework if you just did the full score regression and then you just looked at the, I think it's probably equivalent, to be honest, is if you're using within data, like within sample.
Yeah, right.
I'm going to take one step back, because going on your previous statement that you made about, okay, so you're getting these estimates, they're genome-wide, right? But given how you constructed your adjustment in an MR, shouldn't we go back to the weights themselves and adjust each one? So then in constructing your polygenic, risk or index, that it should be an adjusted weight where everything is now attenuated by a potential assortative mating term.
Yeah, so that's also kind of the kind of dirty nuance hiding underneath it. So I've assumed that the PGIs are in a sense unbiased estimates themselves of the genetic factors. And so my claim is that there is bias, but that the bias is, the fact that we're not doing that adds some bias to the polygenic scores relative to the true genetic factors. But that bias exists in the numerator and the denominator, and therefore maybe we're okay. Or maybe we're close enough that, we see that 15% number that I quoted and say, yeah, it's not perfect, but I think it's like gets us into the ballpark of how big of a problem we think we're actually in. The issue with doing it one SNP at a time, so when you're doing a genome-wide correction, you're working with quantities that are kind of medium-sized. And so we take the ratio of two medium-sized things, and so it's well-behaved. If we were to correct each weight of a PGI separately, then what we're going to do is we're going to add a ton of noise to each of the weights in the PGI, and therefore add a whole, and then we add it together when we construct the PGI, and therefore all the PGI is just going to be 99.
There already is a lot of noise in these PGIs.
And I think you're going to multiply the noise like 100-fold. And so I think that the noise that's induced by ignoring assortative mating in the weight construction and then hoping it cancels out in the ratio is smaller than the noise that you would induce by trying to do it one SNP at a time. That's also kind of the MR estimates are also sort of a composite, so while each GWAS, adjusted GWAS estimate is very, very noisy, an MR estimate is sort of a combination of these things. And we hope the noise cancels out in aggregate.
There's a smaller number of SNPs.
It's also a smaller number of SNPs, yeah. So, yeah, I agree it's a problem. I think it's a kind of just a medium problem.
So then, given that though, would your interpretation of these results be as a lower bound on the correction? On the possible correction, right? Right, like
yeah, that correction should be a little bit larger.
Like if we could perfectly correct, right?
Yeah, and so and that's what we see in the this is MR. This is what we see here, right? So in general, we get almost to the bar, but not quite all the way. And always, it's just it comes a little short, right? So I guess if we wanted to, we could multiply this correction by. My guess is that most of this gap between the dash bar and the actual height is due to within chromosome stuff that's happening. I think it's mostly that.
Yeah, but so are these results using estimates of the PGI, or are these results?
Oh, do you know what? You're right. This is the true PGI. We need to do it.
This is like the best you can do.
This is the best you can possibly do. You're right. That's right.
Right. So I think, it would be, so this is, I think it sets an excellent baseline. But I think it would also be really illustrative to see
how it does when you're estimating.
Yeah, like in a typical pipeline, right? Both either genome-wide PRS or like a pruning thresholding, right? To see really what remains. And I know that those will come with certain assumptions and everything, but at least those can be laid out. Really, and say, here's the gap under those sets of assumptions to get these results.
Yep. That's right. But yeah, so yeah, this is the best you can do, and you think that it's always a little short. So I was thinking these corrections as conservative in that sense. They'd probably be a little more conservative if we used estimated weights instead of true weights.
We are past here. Are there any questions online? Because there will be some time to talk afterwards if people want to join us. But any question from those online? Okay. No. Thank you, everybody. People have been running out, but thank you very much.
Thank you.